I don't see the recording. Anyways, you're recording it. All right, morning, everybody. For us, it's morning. For some of you, it's afternoon. So good day, everybody. And thank you so much for coming to the ZOOMinar today. And thank you for all of you who attended the ECR symposium this past Wednesday and for your support for the young researchers. We had some very excellent presentation. In fact, all of them are really excellent and great. And just want to remind you that our next ECR symposium is coming on July 14, so it will be great to see you again and to give you support for the young researchers. Before I start, I want to remind everybody that we have two speakers today for today's session, and that each talk is about 30 minutes with 10 minutes Q&A session. This will be read from the Q&A posting. And after the second Q&A session of the second speaker, we will be allowing people to come into the panel and ask their questions personally. And just want to move on presenting our first speaker for today. It is my pleasure to introduce Dr. Wenjing Wang. Dr. Wang received her bachelor's degrees in chemistry from Xiamen University of Fujian, China. And then she came to Michigan for her PhD studies, and she did her PhD studies in Michigan State University with advisor Professor Babak Burkham, focusing on rhodopsin mimics for design, rhodopsin mimics design for wavelength regulation of retinal. Then she did her postdoctoral studies in the laboratory of Professor Alicia Ting, first in MIT and then in Stanford. And she worked on developing and designing molecular tools for mapping neuronal circuits. And in 2018, Dr. Wang returned back to Michigan after a 6 years break for her doctoral studies to start her own lab in the Department of Chemistry at University of Michigan, Ann Arbor. And Dr. Wang continues her work on molecular tool design with applications for monitoring and manipulating of multiple biological processes. And I don't want to steal more of her time. So nice to have you here, Wenjing. And if you can please share your screen, we can start your presentation.
Thank you, Meta, for the kind introduction. And also I want to thank Rens for the invitation so that I can speak to you here today. So can you guys see my screen okay?
Yes, we can see it.
Okay, great. Yeah,
it's perfect.
Okay, thank you. Yeah. So I'm going to talk about two short stories of the ongoing research in my group, and one will be related to designing molecular sensors for studying neuronal signaling, and the other one is designing nanobodies for the alpha-synuclein fibrils. So just a quick overlook of my lab's research. So we focus on various molecular sensor tools, and the main approach we use is direct evolution based on protein engineering methods, and the goal is to to study the different neural processes with high spatial and temporal resolution, and for a better understanding of how brain functions. So we have a big interest in designing molecular sensors to allow us to look inside the brain and to see these different signaling molecules being released. So as we all know that, so brain is very, very complicated, right? Made of these billions of neurons that are interconnected to each other. And so the network kind of forms a foundation of how we think and how we behave. So furthermore, in the brain also, there are these small signaling molecules that are being released at specific time and space, and to regulate the firing ability of these neural circuits. So that's very important for modulating neuronal activity. And a major question in the field now is when and where these small molecules are being released. So when and where has a big indication, because dopamine not only makes you happy, but if they are over-released, over-activated in certain brain regions, it can also cause hallucinations. So therefore, it's important to kind of know when and where they are released and what kind of neural circuits have been impacted. Currently, our lab is particularly interested in studying the endogenous opioid signaling. So this is because of the importance of opioid signaling. They modulate various molecular processes such as pain, perception, emotions, like stress response, and so on. Opioids remain the most effective pain medications, but because of these other kind of processes they're regulating, so they also come with these severe side effects such as addiction and also respiratory suppression. So in order to design better pain medications with less side effects, it's important for us to have a better understanding of how endogenous opioid signaling works. So in order to study that, we need these new molecular sensors and tools to allow us to see what kind of stimuli will trigger these peptide release, and also where are these peptide released. So the current state of art for detecting opioid peptides is this fast scan cyclic photometry and microdialysis. So these bioanalytical methods are very powerful. However, the major limitation though is they have very poor spatial resolution. Because they cannot allow us to really look at how these small molecules interact with each neuronal circuit at a cellular resolution. So in the past decade, these genetic encoded fluorescent sensors have been very transformative. They really allow neuroscientists to look into the brain and to see, look at various signaling processes. And so our goal is to engineer these fluorescent sensors for detecting opioid molecules as well. And this work is led by a very talented graduate student in the lab, Kayla Cloning, so who's the first graduate student to join my lab. So Kayla wanted to take advantage of the existing opioid receptor, which can already recognize these opioid molecules to design these sensors, right? So this opioid receptor, upon binding to the opioids, the intracellular side of the protein will have a conformational change. Actually, these opioid receptors are G protein coupled receptors. So it will start recruiting downstream signaling molecules. And for fluorescence readout, so we added a version of fluorescent protein. So this version of fluorescent protein is very sensitive to the environmental change. So upon environmental change, and there will be fluorescence increase. And to cause the environmental change, we added another protein. So this is a nanobody. It recognizes only the activated conformation of the opioid receptor. So that means only in the presence of opioids, and this nanobody will bind to the receptor, therefore causing an environmental change surrounding the fluorophore. And that could cause a fluorescence increase. So once we have this design in mind, in our lab, we just assemble the gene fragments together and clone it. Then we test these in HEK cell culture first. So after introducing these into the HEK cells, and then we will monitor the sensor in the presence or in the absence and presence of opioids. So in the absence of opioids, so there's very dim fluorescence. And upon addition of opioids, we realize so notice something very surprisingly. So instead of immediate fluorescence increase, what we observe is this gradual fluorescence increase. It takes very long time. What we initially imagine is that this conformational change should be very fast. So fluorescence increase should be fast as well. So because of this long duration for the fluorescence to increase, that makes us think that it's probably taking a different mechanism. So one hypothesis we have is, so these fluorescent proteins are fluorescent because So inside the beta barrel, there are these 3 amino acids that can undergo chemical transformations and to finally form this fluorophore, which is can be fluorescent. And this takes like 40 minutes to hours. So it's a slow process. And once this fluorophore is formed, the reason is that it's very sensitive to the environment is because the protonated form of this fluorophore is not fluorescent, but the deprotonated form is fluorescent. So therefore, the pH change of environment, electrostatic environment change could affect the fluorescence intensity. So the question we have now is, how do we differentiate whether the fluorescence increase is due to environmental change, means the deprotonation or protonation state, or whether it's due to the fluorophore maturation. So we have been puzzled by this for a long time because the sensor molecule is a membrane-bound protein with these complex protein components. It's very hard to perform mass spec to determine right, the mass difference. And so finally, we realized actually the solution can be much more simpler because we could just do a simple pH titration. So therefore, we characterize a sensor in the presence that's treated with opioids or without opioids and at different pH. And we did notice that so at higher pH and there's higher fluorescence. At the same time, now we can compare. So at high pH, when the fluorophore should be fully deprotonated, and we can compare the fluorescence with opioids or without opioids. So here's a titration curve that you can see. So in the presence of opioids, so we clearly see higher fluorescence compared to the no opioids case. So that indicates the mechanism is due to the fluorophore maturation. So so we didn't think of this initially because this is a mechanism that hasn't been reported before. And so what we have observed so far is that so in the basal state without opioids, the sensor fluorophore cannot mature. But in the presence of opioids, then there's a conformational change, and then the fluorophore can mature. So what could be causing this like change? And one hypothesis we have is maybe only in the close conformational state and the protein, the green fluorescent protein can fold properly to allow the fluorophore to mature. And one easy way to test this is doing a negative control. So if we remove the nanobody 39, then the sensor will not be able to adopt the conformational change. So what we expect to see is that under this, so in the negative control, there will be low fluorescence. To our surprise, so what we notice is that, so instead of low fluorescence, and we observe already high fluorescence, even in the absence of opioids. So that indicates that, so this suggests that it's the presence of these MB39 nanobody that can suppress the fluorescent proteins fluorophore to mature, which is very surprising to us, right? Because this has never been reported before. So to further validate this, then we made a simpler construct. So just make a soluble construct of owning the this green fluorescent protein fused to these nanobody and also the green fluorescent protein by itself. And we expect to see low fluorescence for the fusion construct and high fluorescence for the fluorescent protein by itself. And so that's exactly what we observed. So this really supports that. So the presence of these MB39 somehow can like disrupt the folding of these green fluorescent protein through some unknown interactions, and that would inhibit the fluorophore from mature. Okay, so so it seems all like very clear point, but it does take us a long time out these unexpected mechanism. So now we have a better understanding of how this sensor works. Is that so MB39 somehow can interact with these version of green fluorescent protein and inhibit, possibly disrupt its structure and inhibit the fluorophore from maturing. And in the presence of opioids, so these nanobody 39 will interact with the opioid receptor, therefore removed from this green fluorescent protein, and then the fluorescent protein can fold correctly and the fluorophore can mature. So a better understanding of this mechanism is important for the further optimization of the sensor. And at this point also, so we gave a name to this new sensor for detecting opioids called Spotted for single-chain protein-based opioid transmission indicator tool. I think this is one of the most fun parts for designing new tools, that you get to name it yourself. And so more importantly, we also want to test whether this sensor can work in neuron cultures as well, because eventually we want to apply it in animal models. So we test this in neuron cultures as shown here. So for the green fluorescence channel, so without opioids, so there's some background, but in the presence of opioids, we see much stronger fluorescence. And we also performed opioid titration. So the sensor shows similar binding affinity to the opioid receptors binding to the opioid, which is good to know. So, and only just last week, and so Kayla has designed an improved version of this sensor with better signal to noise ratio. So we're very excited for the next step for this project. So next we are going to apply Spotted in the animal brain, and to be able to detect the endogenous opioid peptides in the brain. And more interesting also, it'll be interesting to look at where the different synthetic opioids are targeting in the brain. At the same time, so this opioid sensor could also be very useful for screening of new opioid molecules. And so Kayla is now collaborating with the Chemical Genomics Center to develop this into a screening platform. So with that, so that's just a short story showing the sensor design projects in our lab. And now I'm going to change gear and talk about a nanobody design for targeting the alpha-synuclein fibrils. Why do we want to develop nanobodies while there already are a lot of antibodies existing for these protein aggregates? So antibodies, even though are very useful, but they have limitations because these antibodies usually are very large in size and also have very complex structures. They are very expensive and hard to produce. And furthermore, so antibodies, due to the complex structure and disulfide bonds, so they cannot be easily expressed and stable inside in the reduced environment inside the cell. So for this project, we actually want to target the protein inside the cell. And so recently it's found in this llama, a different kind of antibody. So instead of two domain, so it's only a single domain for the target recognition site. And if you extract out the target recognition site domain, and so this is what we call the nanobody. So the nanobody here is very small in size. It's only 1/10 the size of the conventional antibodies. And even nicer is, so it's much more stable than conventional antibodies, and also can be easily expressed intracellularly, and also very easy to produce. You can literally produce it in E. coli bacteria culture. So, and one of the targets that we want to design these nanobodies for is the alpha-synuclein fibrils found in Parkinson's disease, because a lot of these fibrils are mainly formed inside the cell. So we really want to be able to target the pathology development inside the cell. So that's the reason we want to develop nanobodies that can be stably expressed inside the cell. So as I mentioned that, so already there are antibodies that can target these fibrils, but because there are limitations in stability inside the cell and also hard to cross blood-brain barrier, so that's why we still want to develop nanobodies. And also, so there are some nanobodies developed already against the alpha-synuclein monomer, but we want more selective targeting for the fibril form instead of the monomer form, because in normal cells, this alpha-synuclein monomer actually has physiological functions. So using the nanobody that recognize the monomer form could perturb the normal physiological functions. So therefore, the overarching goal of this project is to be able to develop and design these new synthetic nanobodies to selectively target alpha-synuclein fibrils, and we can also express and introduce these nanobodies intracellularly to prevent the alpha-synuclein pathology development. So, so one important part for our synthetic nanobody design that's distinct from the previous work is that because we intentionally wanted to use this nanobody inside the cells, but inside the cells, it's reduced environment, and in the nanobody, there is still one disulfide bond. So that could be reduced in the cytoplasm of a cell. And that might cause some conformational change of the protein. And in order to ensure consistent structural folding, like both outside the cell and inside the cell, so we decided to remove the disulfide bond by introducing two cysteine mutations. So that and so we introduced two hydrophobic residues here because so inside this beta barrel is very hydrophobic environment. So we have validated also by introducing these two mutations, the nanobody can still fold properly. So next is to generate these nanobody libraries and then for the selection against fibrils. So we follow previous protocol for generating these nanobodies. So we keep the beta barrel constant as shown here, so which serves as a scaffold for the nanobody, but we vary the loops highlighted in blue here. So because it's found from these nanobodies that so these loops are variable, like from nanobody to nanobody. And so we just introduced the generated bases into these loops so that any kind of amino acid could be possible to be introduced in these loops. And for the loop 3, we also introduced different lengths, like just following the previous protocol so that to have these different structure folds. And after synthesizing these randomized DNA sequences for nanobody in the lab, then we introduce these electroporate this DNA into yeast cells to create yeast library. So each yeast will pick up one DNA plasmid, and so it will only express one kind of nanobody on its surface, and express on the surface because we fuse the nanobody after a yeast surface protein called AGA2P so that it can be displayed on the surface. And so once we have this yeast library, so the rest of the work is very straightforward and easy. So we can just incubate the yeast culture with the protein target. So in this case, it will be alpha-synuclein fibrils. And then can use antibody stain against the alpha-synuclein to detect the fibrils that's bound to the nanobody. So the detection can be performed either by max activated cell sorting or fluorescence activated cell sorting to select for the ones that shows higher binding to the fibrils. And the enriched yeast cells then can be sequenced to determine the properties of these nanobodies. So here shows the selection scheme for our nanobodies towards these alpha-synuclein fibrils. So in the starting library, you can see we treated with both fibrils or no fibrils as an active control. So in either cases, in the initial library, so you can there's like no binding to the fibrils, and then we just draw a gate to enrich the yeast cells that shows high PFF signal. And as the selection goes, so then we start enriching the yeast populations that shows higher PFF binding, right? So oftentimes also you will get some false positive clones from this selection. What that means is, so you notice that there's one population of yeast here, no matter we add PFF or not, and they show up. So these are nanobodies that can directly bind to the antibody. Fortunately, in this case, so these two populations are well separated. So therefore, we can draw a gate and to select for the population. So that's more PFF dependent. So after these selections, so finally we were able to enrich yeast populations that shows the higher fluorescence in the presence of the fibril. So from that, then we can just like sequence these yeast cells and then categorize them individually. So Mima was able to that identified 28 these fibro nanobody clones, and so here shows the characteristic that representative characterization of these nanobodies. So as you can see, so in the top row is when the yeast cells are treated with the fibro, so they all show high fluorescence signal, and the middle row here is without the fibros, so therefore no binding. So what we are very happy to see that is that, so we also incubated these yeast cells with the monomers. So with these monomers, we don't see binding either. So that indicates that these nanobodies are selective towards the fibroforms. So next step is to further characterize these nanobodies. So we collaborated with Professor Xiaobo lab at Johns Hopkins, and they performed the native page gel Western blot. So by running the monomer and PFF and the fibroform of the alpha-synuclein on this gel, and then if we use the antibody against the alpha-synuclein, so we can see that this antibody cannot differentiate the monomer or fibroform, so it will bind to both the low molecular weight monomer region, and also it will bind to the high molecular weight fibro region as well. But for the nanobodies that we selected, so these nanobodies, they only recognize the high molecular weight fibroform, but not the low molecular weight alpha-synuclein. So this further kind of supported the fibroselectivity of these nanobodies. And we also want to make sure that, because those are recombinant fibroforms, we also want to make sure that this nanobody can bind to the alpha-synuclein fibroform in a slightly more physiological conditions. So then we followed Professor Virginia Lee's protocol to generate these fibros in hex cells. So we generated a stable cell line expressing the alpha-synuclein mutant, which tends to aggregate, and then after transducing the recombinant PFF, so it will start the alpha-synuclein aggregation in the hex cells, and also started the pathology development of this alpha-synuclein. So the hallmark here is the phosphorylation of the alpha-synuclein at position 129. So these hex cells are also affected with the nanobody we developed by fusion to GFP. So what we hope to see is, this pathology development of alpha-synuclein, and also colocalization of this GFP with the aggregates of the alpha-synuclein. So as shown here, so the top row is treated with a fibro, the bottom row is no treatment as a control. So without PFF, so we don't see pathology development of alpha-synuclein. And on the top row, so we started seeing these aggregates and higher amount of these P129 phosphorylation. And we also see some colocalization of the GFP along with this, so which is good to see. So next step is to check whether these nanobodies that in the more physiological conditions can actually inhibit the pathology development. So to do this, so Xiaobo's lab, they performed this experiment by expressing the nanobody in neuron cultures that have the humanized alpha-synuclein, and after transduction with the recombinant PFF, so these neurons will also start developing the pathology for alpha-synuclein. So what we hope to see is with the expression of our nanobody, we hope to see inhibition of the pathology development, but in the control neurons, which only express the green fluorescent protein, and there will be high level of pathology development. So they tested this experiment. It's actually quite to our surprise that it worked very well already. So in the top row is a control. So as you can see that there's high level of pathology development, while when the neurons are expressing nanobodies, and we see a much more reduced this pathology development. So that encouraged by this, then we move on to testing this in animal models. So in animal models, so very similar procedure, except that we use the mice So these AV viruses expressing these nanobodies are injected into the newborn mice, so that we can get a wider spread of the AV infection. And 2 months after, then we inject the PFF into the mouse brain, so to start the pathology development. And after 1 month, typically the whole brain, so the pathology will be spread to the whole brain. Then we sacrifice mouse and then look at effects. So this is one of the cortex region that we look at first. So similarly, we have the control animal, which is expressing only the GFP, and you can see there's high level of pathology. But for the animals that are expressing our nanobodies, we also see a much reduced pathology marker. And we look at other than cortical cortex region of the mouse as well, and observe similar things, a significant reduction of the pathology development. Actually the AV injection, it didn't spread to this deep brain region. And so in these regions, there's no green fluorescent protein expression of our nanobody expression. So in these regions, then we do not see a difference between the control animal and our animals. So all of these really support that. So the expression of these nanobodies intracellularly can inhibit the pathology development of this alpha-synuclein. So we're very excited about this, and next is to see whether to perform behavior studies to see whether expression of these can indeed improve the cognition of these animals in these animal models. And so far what I have seen here also is the nanobody is expressed before the PFF transduction. So it works more like as a preventative method, but we're also curious to see if we introduce the PFF first, is it possible to use these nanobodies to degrade the fibril that's already formed? So next step is to add like degradation tag to these nanobodies and to see whether we can reverse the pathology development. So with that, I want to thank Dr. Yamima Rani, who performed all the nanobody work, and also Pilar Cloning, whose work was shown in the first part. Yeah, so here's a picture of the group, and so the students, they're really fun to work with. And I guess with that, I'll be happy to answer any questions.
Thank you very much. Such a great stories.
Thank you.
There are lots of questions that are coming up, and a couple of the first ones are from anonymous attendees, and I'm going to read them. It's what were the cysteine mutations? I assume this refers to your nanobody. And then the second question from, let's answer this one and I'll just go on the second question.
Yeah, okay. So here shows the cysteine mutations. So we literally just mutate the two cysteine, like one into a leucine, one into an alanine, so that they can, so we did a very simple kind of modeling so that they can occupy similar kind of space as the two cysteine residue.
Okay. So the next question is from Vinda Sand. The question is, what type polymorph of preformed fibrils we use for FACS? Are the nanobodies blind to polymorphism?
Yeah, okay, that's a good question. So we actually get these preformed fibrils from our collaborator, and so we follow a protocol of like so it's preformed fibrils, and then we sonicate for about 30 seconds and to get these like smaller fibrils. It is like heterogeneous, and so are the nanobodies binding to the polymorphism or not? So we haven't looked too much into this yet, and to see that would be, yeah, that's a very good point though. So that would be next thing actually we want to study to see, because we have 28 of these nanobodies, and we want to see whether they will show different kind of binding affinity for different forms of the fibrils.
And the next questions, you got a lot of questions from Christian Grissinger. And I tried to promote him to the panel, but so he can ask them, but I'm going to read them to you. Oh, here he is. Hi, Christian, can you ask your questions? I don't know, he's still muted, so I'm going to
okay, yeah, I can read the questions here.
Yeah, so this is do you know what the affinity of the nanobodies for the alpha synuclein fibrils, what concentrations of nanobodies one can achieve in brain?
Very good questions. So we are so we are in the steps of categorizing the affinity of these nanobodies for the alpha synuclein fibrils. I would think they are probably in the low micromolar or high or sub-micromolar range, but that still needs determination, and which kind of just, so that's a much harder procedure for our lab, but the animal, I want to make sure actually they have some effect in animals first before any further categorization, and what concentration nanobodies can one achieve in the brain. So we deliver these nanobodies currently using the AV viral expression system, and depends on what promoter you are using. So you can get like different concentrations. I think with a normal promoter, we can easily get like tens of micromolar concentration in the neurons, but it's intracellular. Yeah. And right, so eventually we do want to deliver these nanobodies like by injection into the blood, and possibly by modify the nanobodies so they can penetrate the blood-brain barrier easier. Okay, the next question Christian has is...
Yeah, Christian is in the panel. Christian,
yeah, hello.
Maybe it will be the easiest, like you have one more question, and after that you can ask them on the free discussion.
Yeah, it took some time to get in, but now I have no access anymore to the questions and answers. So
okay, I can.
Now I forgot my question, but I have another. I think I have another one, which is.
Yeah. So you asked what type of mice did they use in the animal experiment? Yeah.
Yeah, exactly. Yeah, the wild type mouse.
Yeah, it's not wild type mouse. So it's a humanized mice. So these mice are expressing the human alpha synuclein because the nanobody will develop for the human.
And is it are they overexpressing or just normal expression level?
That's a good question. Actually, I'm not sure. I'm not sure whether it's overexpressing. It might be overexpressing. That's why the pathology developed really fast. Usually they can achieve, it can spread to the whole brain in a month.
Okay, could I ask another question, which is, I mean, what's the mechanism of uptake of the nanobodies in the cells? How does that work? Yeah,
so currently we are expressing, so we introduce using viral expressing system to express these nanobodies. So they are
okay.
Yeah.
Okay. So they are already in the cell because they are
right.
Okay. Okay. So
right.
you don't apply the nanobodies from the outside of the cell, hoping then that they go in.
Yeah. Right. So we haven't tried that yet, but that would be the next thing to see. Right. But using AV is much easier. So right, that's what we perform first.
Okay. Thanks a lot.
No problem.
Yeah, Christian, you can stay too if you have more questions. I'm going with one more question because I don't want to take that much time from the other. Maybe a couple more questions. It is from Ashutosh Tiwari, and he says, or she says, very nice work. Have you carried out stability studies on the nanobodies after substituting with hydrophobic residues? How long they're active at 37 and so forth. So how they compare stability-wise and solubility-wise compared to the ones with the cysteines?
Yeah, so that's a good question, right? So the problem is it has to be determined nanobody by nanobody. It's cannot be applied to like all. But what we have noticed is so without disulfide, the nanobody is slightly less stable compared to the nanobody with disulfide bond, because we have performed binding assay with a well-known nanobody, the GFP nanobody. And when we introduce these mutations, so the binding affinity decreased a little bit compared to the original with disulfide bond. But the nice thing about these disulfide bond-free nanobodies is, so we know we can express them in the cytoplasm of bacteria, while for the cysteine, for the nanobody with the cysteine, you have to express them in the periplasm, which is the oxidizing condition. Yeah, but regarding the thermal stability and so on, we haven't performed that study yet. But my intuition is that it's going to be less stable compared to the cysteine, because cysteine disulfide bond usually stabilizes proteins, and by mutating them away, for sure, could affect the stability a little bit. But although we gain these consistent folding in both oxidizing and reducing conditions.
Yeah, great, thank you. So that's the last question.
Okay.
Which is coming from the anonymous speaker. It's more about on which species do you think your nanobodies act on? And I'll just read the full questions. Nice talk. Do the nanobodies target fibril ends or fibril surface? Can nanobodies inhibit fibril elongation or secondary nucleation, or changing the aggregation kinetics of alpha-synuclein? Does the fibril morphology matters? You answer that. Thank you. So just if you can answer the first questions.
Yeah, that's a great question. That's something that we want to understand as well, to see what's the mechanism for this. So what we can predict is these nanobodies probably bind to the C-terminal end of the fibril of the alpha-synuclein, because they are rich. A lot of nanobodies we found are rich in these positively charged residues, while the C-terminals of alpha-synuclein are rich in negatively charged residues. And so probably the nanobody inhibit fibril elongation, secondary nucleation. So we still don't know the answer yet. We tried to perform this experiment once, so 2 times, but we get inconclusive answers. So I couldn't really answer this question now, to see how they prevent the fibril formation.
Okay.
Yeah, that would be really interesting to know. Yeah.
Yeah. This is down the lines, but we will be discussing that further. What are your thoughts about the toxic oligomeric species and your nanobodies? But we'll leave that for the discussion. And thank you so much. And we can proceed to the next speaker.
Yes. Hello. So it's a pleasure to me to introduce Tessa Sinha. Tessa studied at Utrecht University at the Bijvoet Center. So she did a master's studies there with Stefan Rüdiger on protein folding and the action of chaperones. Then for a PhD, she also did at the Bijvoet Center, but then with Mark Baldus, working on solid state NMR on membrane proteins, and she also worked on NMR method development. Then for her first postdoc, she moved to Cambridge, working with Chris Dobson and Michelle Vendruscolo. And there she started to work on C. elegans, and also using fluorescence lifetime imaging, and focusing mostly on amyloid beta. And then for the second postdoc, she moved to Northwestern University, to the lab of Rick Morimoto, focusing on in vivo of aggregation of polyglutamine expansions, again in C. elegans. And then last year, and I was very happy about that, she was offered a faculty position at Utrecht University to start her own group. And I'm really glad she accepted and has joined again the Bijvoet Center, where she now has set up her own lab. And now I would like to give the word to Tessa, and so she can tell you more about her work. Tessa?
Thank you very much, Antoinette. Let me share my screen and get the laser pointer up. Okay, can everybody see this?
Yes.
Yes. All right, so thank you again, Antoinette, for the introduction, and thanks to Magda and Rams and the other organizers for having me in this series. So today I will talk about my attempts to understand amyloid formation in vivo in C. elegans, specifically focusing on polyglutamine aggregation. And so most of what I will be telling you today is my postdoctoral work in Rick Morimoto's lab. But at the end, I will also come to some very preliminary data from my own lab. Sorry, my slides are stuck. Okay. So basically, we are in the proteinopathy seminar series here, so I don't really need to introduce this topic in too much detail. But I would just like to point out that this is a really widespread phenomenon of amyloids like protein aggregation affecting different cell types and different tissues in many different diseases. But I'm mostly interested in those that are neurodegenerative. So here you see Alzheimer's, Parkinson's, and Huntington's, where the brain tissue really shows these micrometer-sized insoluble deposits. And when you look at these under the electron microscope, you actually see that these proteins form fibrillar structures. So the question is now, how do you try to study this process, and mostly the kinetics of it? So how do these fibrils actually come about, and how are these insoluble deposits growing or potentially spreading? This is something that, of course, by many of us has been studied in great detail in vitro. For example, using thioflavin T fluorescence to monitor amyloid formation. And you can do this really in a very well-controlled system with purified protein or peptide. And even when you do this carefully for a range of protein concentrations, you can then use a set of equations to actually extract all the different steps that are going on. So from monomer to fibril, but also secondary processes and oligomer formation. And you can even quantify the rates of these different steps. So what I'm showing you here is work from the groups of Thomas Knowles, Michele van Ruskelo, and Chris Dobson in Cambridge for amyloid beta. And so this is really very powerful. But the question is, how does this translate to the situation in vivo? So we don't actually know whether these same steps of amyloid formation are occurring and whether the rates are also preserved, or whether the cellular and organismal environment really has major effects on these processes. I'm sorry, I'm really having some technical difficulties here today, but I hope you can still see my presentation. Okay. So yes, the problem is that there's a pretty big gap between in vitro reactions and the human brain. So the way that I'm trying to address this is by using a simple animal model, which is C. elegans, which is a multicellular animal. But at the same time, it is also very easy to work with in a lab, and it's amenable to many of the similar techniques that you would do in typical in vitro assays. And so I think I'm probably the first speaker in this series to really give a C. elegans talk. So I thought I might introduce the organism to you a little bit more in detail. So the adult worm has exactly 959 somatic cells, yet these are divided over different tissues. So the worm actually has an alimentary system composed of the pharynx, which is sort of like the mouth, and an intestine. And it has also muscle cells, it has a nervous system. And so it's really a complete animal, yet at a very small scale. So also we have genetic tools available. So we can make transgenic worms pretty easily that express the protein of interest. So for example, an aggregation-prone disease. Then the worm also has a short generation time. So in 3 days, you actually get from this egg to the full adult. So over the weekend, you can basically grow as many worms as you like. And then also the rest of their lifespan is relatively short. So in about 2 to 3 weeks, they age and they die. And so this means that actually you can study age-associated processes on a relatively short timescale. Finally, the worms are also optically transparent, which is very convenient for our experiments. So you can already see from this top image that you can actually look inside the worm and see all the different tissues. So you even see, for example, the eggs here, and also you see here the pharynx that is depicted in green in the cartoon. So this is really nice. And this also means that when you actually express your protein of interest with a fluorescent tag, you can really follow it in living animals. And so one example of this is the C. elegans system to study polyglutamine aggregation. So this is work from the lab of Rick Morimoto from already quite some time ago, where different repeat lengths of polyglutamine are expressed, fused with yellow fluorescent protein for visualization. And these constructs are expressed in the muscle cells because these are the biggest and the easiest to visualize. And also toxic protein aggregation is associated with a paralysis phenotype when the muscle cells become impaired. So this has been used extensively in the Morimoto lab and also more widely in the field. And so what I think is really cool about this system is that when you go from 35 Qs to 40, you are actually crossing the threshold that also causes Huntington's and other diseases in humans. And you start to see that the protein aggregates. And so the longer you make the Q stretch, the more and the faster you see aggregation. So this really seems that at least to some extent, this process is conserved between what we know about the properties of the polyglutamine protein and the biological environment of the C. elegans muscle cells. So, okay, these worms are nice to look at, but now, of course, we want to use them to really obtain a mechanistic understanding of how these aggregates are developing in a living animal over time. And the main question in this project that we asked was whether we can also apply the same kinetic methods that I showed you earlier that are used in vitro. Can we apply these actually to data coming from in vivo and do the same mechanisms hold true? So I started out with these Q40 YFP animals because they show really nice, robust inclusion formation. So you see here different time points during the life of the animal. And the protein is expressed in the muscle cells as before. So you see a very nice outline of the muscle cells. And at early time points, all of the protein is diffuse. But then as the animal is getting older, you see more and more of these bright spots, which we call inclusions, appear. And finally, all of the protein actually localizes to these inclusions. And there's no more diffuse protein visible. So as I showed you in the introduction, I am interested in amyloid-like protein aggregation. So is this actually amyloid, or is this just somehow protein precipitation? And to address this, I performed electron microscopy on sections of the worms. And you see very nicely the inclusions because they're really micrometers big. And they are localized just below the sarcomere. And then when you go to higher magnification, you see this spaghetti-like structure that is indicative of fibrillar polyglutamine. Now, another hallmark of amyloid formation that we know in vitro is the concentration dependence. And so we also wanted to check whether this was the case in our system, or whether it would, again, just be like crossing some sort of threshold where the protein precipitates. But we found, in fact, that it was concentration dependent. So the trick we used here was to compare homozygous with heterozygous animals, because the worm has 2 copies of each chromosome, just like we do. And so this means that you can take the wild type or the polyQ animals, cross them with wild type animals, and you will get heterozygous that have only half of the number of copies. And this results in a very clear difference in the aggregation kinetics. So we are simply counting here the number of inclusions, so these bright spots that I showed you before, in a population of animals as they are getting older. And you see a clear difference in the lag phase, and also the heterozygous are more slowly to reach the plateau. But what was a little bit surprising at first to us is that we actually get to the same total number of inclusions almost. So it's not like when we have half of the protein concentration, we also get half of the number of aggregates, which we had maybe naively assumed. And after repeating this experiment several times, I realized that we always get to roughly 100 inclusions. So I thought, well, this has to mean something. And indeed, it actually means that this is the same as the number of cells that we are expressing the protein in. So there are 95 of the so-called body wall muscle cells. And then there are a few other muscle cells in which also we observe some expression. And so this number of inclusions is actually very close to that number. So it means that the protein concentration changes the kinetics, but always results in, on average, 1 inclusion per cell. So that means that actually we can also relabel our axis here. And basically what we are looking at is the number of cells that have acquired their inclusion. So this is a little bit mysterious. You might wonder, why do the cells not all undergo aggregation at the same time? And this picture here was taken at exactly the half time of the aggregation reaction, if you will. And you actually see that some cells have already acquired their inclusion and other cells are completely yet unaffected by aggregation. So also what you see from this picture is that these cells actually all look the same. I mean, they have roughly the same size and shape, and we know that they also have the same physiological function. So we did some test experiments to show that actually all of these cells have the same aggregation probability. So it's not like some are more prone to aggregate, and then that's why they would be earlier. So the way we checked was that first, I examined where in the animal the first inclusion would appear. So you see some examples, and I have many more. But basically, the first inclusion is always somewhere random along the axis of the worm. And the asterisk is labeling the head. So it's also not dependent on whether it's closer to the head or the tail. It's essentially random. Another thing we considered was that maybe the aggregates are spreading. So maybe there's one random cell where aggregation starts, and then it actually spreads to other cells. So that's where this time dependence comes from. But this did not seem to be the case either, at least not from these observations, where when we look at animals that have just a couple of inclusions, there is basically no spatial correlation between them. So at this point, we also ruled out this explanation. And then to make a long story short, the answer to this question of why the cells do not all aggregate at the same time is because nucleation becomes stochastic in small volumes. So what do I mean by that? Well, nucleation we define as the first misfolding event that leads to aggregate formation. And so nucleation is always required for aggregates and amyloid fibrils to form. However, when you look at this in a bulk in vitro reaction, you basically have many molecules in your solution, and the aggregation behavior is going to be deterministic. So if you repeat your experiment many times and you are careful enough with the conditions, you're basically always going to get the same numbers of nucleation events at these different time points. Now, what people have also shown in vitro is that when you break this down into nanodroplets, the aggregation behavior becomes stochastic, just because there are very few nucleation events, especially when you look at the scale of the single droplets of like femtoliter to picoliter size. And so another way to explain this stochasticity is when you look at an individual droplet in this system, you basically cannot predict where or when nucleation is going to happen in that particular droplet. And so this, we realized, is actually quite similar as the C. elegans system, because we have about 100 muscle cells. Here I'm only showing you 25 for simplicity. But basically, also in this system, we expect stochasticity because the muscle cell volume is 1 picoliter. So basically, even though nucleation events are happening over time with a certain rate, we do not actually know when it's going to happen in which of the cells. So this is actually the explanation for the behavior that we see. So this basically then led to the initial model of what's going on in this system. So at the level of the single cell, you have stochastic nucleation. And after that, directly the aggregate can grow. And it will basically grow until all of the soluble protein in that particular cell is depleted. So at the level of the animal, you will see more and more inclusions appearing until basically nucleation has happened in all of the cells and they have all grown to reach their maximum size. So this was an interesting model because it also gave us something to test. Namely, then we should be able also to observe this aggregate growth in the individual cells. And it turned out that we can actually see the growth happening under the confocal microscope. So these animals are alive, but they are just anesthetized. And you see two different close-ups at the top and the bottom at various time points. And the inclusions with the yellow arrows are actually the ones that can be observed to grow over time. So in particular, the ones that are very small at the beginning, you see that they grow bigger and bigger. And also here at the bottom, these two, you can see really by eye already that they are growing. And another interesting feature of these images is that you also see inclusions that are not growing. For example, the big one here. And this actually also does not have any soluble or diffuse signal surrounding it. So it means that this one has already acquired its maximum size and just taken up all of the protein that was around in that particular cell. And also we saw that the growth is actually very rapid. You can already see from my time points that this is happening on a scale of only a few hours at a rate of about 0.5 micron per hour. And the maximum size is usually 3 to 4 microns. So it means that in a matter of hours, the inclusion has reached its maximum size. And so nucleation is really the rate limiting step here. Then before we continue, there is another question that actually was raised already from this initial data set of just simple inclusion counting. So those of you who are doing thioflavin T kinetics might recognize that this looks a little bit like the sigmoidal shape that you often get for in vitro reactions. And this is suggestive of cooperativity, meaning that once you have started forming some aggregates, actually the aggregation rate keeps increasing until, of course, you run out of protein and you level off towards the plateau. So I'm saying here apparent cooperativity because we realized that actually there was something else going on. And we almost forgot that we are, of course, not doing this in vitro, but in a living and developing animal. So the rate or the time frame of aggregation is actually happening mostly during development of the animal. So when you start out from the egg, the animal grows quite a lot. So I experimentally determined at different time points the cell volume of the muscle cells, and it really increases almost 50-fold from the embryo up to the adult. And now our theory of stochastic nucleation predicts that when you increase a cell volume and you keep the protein concentration constant, you actually increase the probability of nucleation, simply because there are going to be more molecules around. So my colleague Georg Meisel, he played around with these numbers, and he actually found that you could reproduce this sigmoidal shape of the curve pretty nicely by just assuming that there is a constant nucleation rate per cell volume, but then taking into account this, this increase in cell volume during development. So this was, in a sense, maybe a little bit disappointing. And also it was annoying, right? Because actually we do not want this change in volume to affect our data. So I will get back to this later, how we solved this, this problem. And then now finally, I will turn to the question of can we do basically the same kind of kinetic analysis that people have done in vitro? So can we basically take our hypothetical model as the basis and set up a mathematical equivalent to try to model this and quantify it. So for this, let me go back for 1 second. In vitro, of course, you can easily get different protein concentrations simply by making different dilutions of your stock solution. And this we now had to do in vivo. So that's, of course, easier said than done. But I also told you already that we have genetic tools for the worm, and we can make different strains very easily. So the trick here is that we usually make use of overexpression. So we inject plasmid DNA into the gonad of the adult worm, which is where the progeny is going from. And basically the offspring is going to take up a variable number of copies of this plasmid DNA. So basically there is no way to predict how many copies or how much protein expression there's going to be. But if you just select enough worms, they are basically all going to be different at this stage. And then you can make stable integrants, and the expression levels will not change anymore, and will be the same within the population of, for example, this one animal that you have selected here. So that's what we did. And I ended up with 4 different strains that had very nicely separated intracellular protein concentrations. So they're here referred to as line A, B, C, and D. And you see also the quantification of the intracellular polyQ YFP concentration. And now these are actually much lower expression levels than from the strain that I was using in the beginning, meaning that we actually don't see aggregation during development anymore. So in the small cell volumes, basically the nucleation probability is not large enough for it to actually happen. And so only at day 1 of adulthood, most of the protein is still soluble. We start to see the first aggregates. And as we let the animals age, we actually get more and more inclusions. And this is luckily still concentration dependent. So you see that depending on the protein concentration, you get less buildup of inclusions. And these are just example images. So I recorded them for 20 animals per day and strain, and quantified the whole data set. And then we were ready to actually do the mathematical modeling. So the first model that I will show you is just pretty simple, assuming there's no cooperativity, so no communication or spreading between the cells. And mathematically, this is basically just what I just said, put into an equation. So we define the fraction of cells that has acquired an inclusion by the small f. And this changes as a function of the nucleation rate constant per cell times the monomer concentration with the reaction order in the exponent. And then times 1 minus f. So that's the fraction of cells that has not yet aggregated. So they are still available for this to happen. And then we define the big F as the actual number of cells. So we multiply by the total number of body wall muscle cells. And we have now integrated this, of course, to get to the expression for the number of cells. So this model basically fits pretty well to our data. And from the fit, we can extract the nucleation rate as well as the reaction order. So nucleation rate is a little bit difficult to interpret at this point, but of course it will be very easy to compare now different conditions now that we can actually quantify the nucleation rate. And I will get back to that also at the end of my talk. And the reaction order is already quite interesting because this is 1.6. So the reaction order basically is of course a measure of the concentration dependence, but also it gives us a number for the minimal nucleus size. So this is basically closer to 2 in our case than 1. So it means that the nucleation process is for sure more complicated than just a single monomer of protein misfolding, which was actually what people found in vitro for longer Q lengths. But at this point, we do not yet have more information about the nature of the nucleation process. So just to illustrate whether or not this is a good fit, I will also show you a not so good fit. And that's for the other model that Georg developed, which was to include cooperativity. So basically, if there would be spreading between the cells, or if there would be a proteostasis effect or an effect on the health of the animals, that the more aggregation they have, the more they become prone to it, then you would actually get this additional term in the equation, which has a different rate constant, and is now dependent on both the fraction of cells that have already obtained their inclusion and the ones that are still available. And basically you see that this does not represent our data at all. And especially for lower protein concentrations, you would really get a huge lag phase, which actually we don't see in our case. However, it's still very interesting that we developed this model, because if we do have other situations in the future where we have aggregate spreading, or we have a big effect of the proteostasis decline during aging, then actually this model would probably represent the data much better. So I would like to come back to this point of stochasticity before finishing this part of the talk, because I told you that this plays a role at the level of individual cells. But actually, I wondered if maybe it also plays a role at the level of the animal, given that it has only 100 cells. So could it be that if you look at one animal and another animal, basically they might have a different number of inclusions simply because of this, this stochastic effect? And actually, this turned out to play a pretty big role. So what you see here is a simulation of the standard deviation in a population of animals. And this is simply based on the nucleation rate that we determined and the number of cells. And so those are the solid lines for the 4 strains. And then the points are my data points for the experimental standard deviation. And you see that actually what we expect from the stochasticity is quite a big part of my experimental standard deviation. So especially for the lowest expressing line, at the first couple of points, it's really actually a perfect match. And only when the animals get older, it seems that there is some more variation coming in. So of course, there are different sources for this. It could be measurement error. It can also be biological variation. But the point here is that previously people mostly attributed this to solely biological variation, whereas actually you have to be careful with that, it now turns out, because simply the limited number of cells also induces stochasticity in this aggregation behavior. So I will conclude this part before showing you some preliminary data. Well, basically the take-home message, if there is anything I would like you to remember from this talk, it's that in vivo protein aggregation kinetics actually works. Of course, this is a very simple model system in which we demonstrated this, but it's the first example. And what we then found using this approach was that the mechanism by which polyQ is aggregating is that there is stochastic nucleation in individual cells. So the cells actually do not communicate with each other in this respect. And once nucleation has happened, there is very rapid aggregate growth, which really absorbs all of the protein in the cell. And so basically we can say that in this system, protein aggregation is governed by the same biophysical principles that we would expect from in vitro. So maybe surprisingly, we do not see any effect of, for example, aging, which would lead to an acceleration in protein aggregation when the animals age. But we do not see this. And it will be very interesting, of course, to find out the reason for that. For example, it could be because we are using overexpression. So maybe just the intrinsic nucleation of polyQ is dominating over other biological effects. So this is something to figure out in the future. Well, currently what we're doing in my lab, one question that we are trying to address is, does this also work in neurons? Some of you might have already wondered about this because, of course, all I showed so far was muscle cells, whereas neurons are the disease-relevant tissue. So the story here is much more complicated. So here you see an image of part of the C. elegans head region, and you can already see that this does not look as neat as my muscle cell images. So the worm has 302 neurons, so it's still like a reasonable number. But of course, they're all different. There are many different types. And also in terms of their size and shape, they are going to be different. But at the same time, we can still recognize inclusions as bright spots. So the arrows here are pointing at some inclusions. And you can already see that now they also differ actually in their size and shape. But we did some preliminary analysis of the inclusion numbers, similar as what I showed you before for homozygous and heterozygous animals. The last few time points will be recorded next week. But what we can already see is that actually the behavior overall seems similar to that in the muscle cells, in the sense that we start from almost no inclusions in young adult animals, and we go straight up. So again, there does not seem to be a lag phase or significant cooperativity. We see a small difference between homozygous and heterozygous, maybe not as pronounced as before, but certainly there appears to be a difference. Finally, what is notable now is that our final number of inclusions, about 10 to 12, is actually very different from the total number of cells, which I said are about 300. So we actually get nowhere near one inclusion per cell. And this could be explained by a subpopulation of neurons being present that are actually particularly vulnerable for inclusion formation. And this has also been suggested in previous literature. So we are currently also trying to find out which neurons those are, and if we can maybe get further with quantifying this system by just focusing on that subset of neurons, rather than having to take all the different aggregation propensities of the different neurons into account. And the last bit of data that I wanted to show you is that, of course, now that we have a model and we have a quantification of polyQ aggregation in the muscle cells, we can look at the effect of basically any gene that we are interested in. So in this example, we are looking at a gene called MOAC4, which stands for modifier of aggregation. This was discovered by the group of Alan Nolan several years ago, and it's conserved throughout evolution. So humans also have homologs of this MOAC4 gene. And interestingly, it actually promotes aggregation. So when you delete MOAC4, which you see here with the dashed lines, you actually see a slower buildup of inclusions in all of these cases. So we are still in the process of quantifying this and performing additional experiments, for example, to figure out the stoichiometry. But we can already see from this that the nucleation rate is definitely reduced without MOAC4. And there are many other genes, of course, that we are interested in studying in the future. Also, for example, chaperones, as you can probably imagine. So with that, I come to the acknowledgements. So I would like to thank Greg Morimoto, who was my advisor during my postdoc for the project I just presented. And we collaborated with the University of Cambridge, where I was a postdoc before, with Michele van Ruskelo, Thomas Knowles, and his postdocs, Gerrit Meisel and Thomas Michaels. I would like to thank Alan Nolan for the collaboration on the MOAC4 project, and of course Antoinette for her support in setting up my lab at Utrecht University. And then finally, I would like to thank my current group, also known as the Worm team. And so here you see us in the lab at the Safe. And here are Jelle and Vera, whose data I just presented. And thank you for your attention.
Thank you very much, Tessa, for a very nice presentation. I see there are a few questions in the Q&A. Shall I read them to you?
Yes, please.
Okay, so the two questions from Gunilla Westermark. Thanks, this was a very nice, interesting presentation. Are the aggregates membrane enclosed?
It seems like they are not. So in the electron microscopy data, we do not see any signs of membranes surrounding them, no. But this is just a relatively simple polyQ construct. So it doesn't have the flanking regions of the Huntington protein. So that's why maybe we also don't expect membrane interaction, but for Huntington, it might be a different story.
Okay. Second question, have you expressed the protein without the GFP?
No, because then we cannot see it. So yeah, I agree that of course the GFP tag is, or YFP in this case, may influence the aggregation behavior. But yeah, that's what we are basing our studies on. So
you can do the staining, right?
Yes, I mean, yes, you can. But I haven't done it. Yeah.
Oh, okay. And a question from Mohammed. Is there any evidence of extracellular aggregates in C. elegans?
Yeah, that's a good question. So it can definitely happen. But yeah, it's not, I mean, C. elegans doesn't have, for example, a brain as complex as humans. So even for amyloid beta, it's not really clear in the current C. elegans models whether it's actually secreted and whether the aggregates are extracellular or intracellular.
Okay, I see no other questions at this point. So shall we move on to the discussion then?
Yeah, I promoted Sunil, who raised his hand. So Sunil, can you please ask your question?
Can you hear me? Hello?
Yes.
Oh, yeah. So, hi, Tessa. I have actually a question from both of you guys, Wenjing as well. Nice presentation, by the way. So first, Tessa, so two questions. So obviously, you guys have done a lot of work with the studying these aggregation kinetic profiles of A beta, I guess, in Knowles group a lot. And I was wondering, I guess you trying to figure it out with polyQ, but I was wondering how much different is it in terms of the aggregation profile, because as we know, with I assume with, for example, alpha-synuclein, it doesn't apply to the same aggregation profile. Like if you include, you know, how your group has came up with these models, which you can fit and tell if it is primary nucleation, secondary nucleation, but that doesn't apply. It's not a generalized approach. That's what I'm coming to. And so I was wondering how much different it could be for the polyQ. That's the one question, yes. And I have another one, but that's, yeah.
Okay, let me first answer that, because this is a great question. So I have also looked at amyloid beta and alpha-synuclein in the worms. And I can already tell you it's totally different for each protein. So for alpha-synuclein, we find actually inclusions that are not amyloid-like for a very long time. They are dynamic, and we have some evidence that this looks like liquid phase separated condensates. And only when the worms are very old, then these like convert into an amyloid state. So that's a totally different process. And then for amyloid beta, we actually see very many small deposits in each cell rather than one big aggregate. So there it looks like there are actually more nucleation events happening in one cell. So we have not yet quantified that, but it looks like the nucleation rate is going to be higher.
Okay. And the other question is related with the number of copies. I guess you mentioned it, that you guys have, for the polyQ, obviously, you picked the worm. So I was wondering what approach, like how much overexpression, like how many copies you think would it be per cell? Is it one copy, or I guess you mentioned at the beginning, but then you guys said that.
Yeah, it's going to be, it can be hundreds of copies. So we don't know. But people assume or have estimated that when you overexpress a protein by plasmid injection, that the worm can take up like 100 to 300 or so copies of the plasmid.
Okay, and so it's a random bombardment, like in the cells. Is that the one approach, or like you put a lot of copies in the vector, like plasmid?
No. So what happens is that we inject into the gonad.
Okay.
And there are nuclei floating around freely that will develop into the embryos.
Okay.
And they actually absorb these plasmids, and they somehow ligate them together. So they build like an extra chromosomal array that contains a certain number of copies.
Okay, so it would be, let us say right now it's fine, you are doing this, but let us say if you have to in future, somebody else is trying to make this strain, and it might, let us say the number of these polyQ protein, for example, out after 40, which aggregates, if they have different number of, would you envision that a different profile, or it wouldn't change?
Well, this again is a super cool question, because it is possible to make single copy gene insertions. So I have also done that, for example, for amyloid beta, and other labs have also done it. Then typically what you see is no aggregation at all. So it seems that we have to be somewhere in between. And so it would actually be very interesting to titrate this down to a level where you see hardly aggregation. And then maybe you would only see it in aged animals. So then you would indeed also get a different profile.
Okay. Yeah. Sounds good. That was good.
Very good questions. Thank you.
Can I go ahead, Magda, for Wenjing as well? I had a bunch of questions with her. I don't know.
Yes. And after that, Christian's turn. Yeah, so, go ahead.
Hi, Wenjing. How are you? It was a nice presentation. I don't know if you remember, we met a couple of years ago in this meeting in Keystone.
Oh, now I remember. Yeah. Nice to see you.
Okay, so I don't know, they blocked my video somehow. They are not letting me see my face, but anyways, so I was wondering if so with your these antibody, very cool. So you said you guys are predicting, and there were a lot of questions about where it's binding, what stoichiometry, what binding affinity. So you saying that they might be binding the C terminus, these antibodies, and a lot of papers right now coming out with the for example, chaperones, they interact with to regulate the aggregation and the function of synuclein. They interact with the N-terminus, and which is apparently supposed to be the nucleation site where aggregation starts. So do you think, so these antibodies, you guys are you are just targeting the preformed fibers which dissolve them, and they have no role, or you don't think they will be influential in kind of inhibiting the primary nucleation or early aggregation process? Is that how, with that hypothesis you guys are going with?
So Niels, thank you so much for for providing that the other possible hypothesis. So we really haven't probed too much into the binding mechanism yet. So ideally, yes, we'll be able to make these truncated fibrils, and then to see and to look at the binding affinity of the nanobody against these different forms, and to see where it binds to. So or perform other kind of, I guess, biophysics, IMR studies, and to see where which part it interacts with. But yeah, so that would be interesting things to look at next.
Okay. And the other thing is, like, there are these, for example, there are some antibodies which are for imaging processes, like A11, and there are some specific for not A11, but there are other antibodies specific for alpha-synuclein. So you guys are also planning to use them as like other than dissolving these fibers, as like a staining tool as well, or this is just for the for kind of more therapeutic medicinal purposes? So
at this point, it's more for just for therapeutic purpose or as a research tool, but not for immunostaining. So the nanobody we have, the binding affinity towards these fibrils are not very high yet. We are thinking they're probably, with more in the micromolar range. So that's what we use for the e-surface display as well. But the antibody, so the one limitation of this nanobody is that compared to antibodies, which have two domains, so it's much easier to get high binding affinity antibodies than nanobodies. It's not impossible, but we only did perform one round of selection. So so ideally is that, so we can use the nanobodies that we have selected and randomize them and perform another round, maybe even a few more rounds of selections to get even higher affinity nanobodies. So then that would be good enough for understanding. Although the way I see, because already there are antibodies that can do that, so it's not going to be much more advantageous to have. Of course, it can. It's much cheaper to get these nanobodies, but for us now, it's more interesting to see the intracellular expression profile and how they can inhibit the this aggregate formation.
Okay.
But those are very good suggestions.
Okay, well, good work both. That's really nice. Very extreme, like different projects, Thank you very much.
Thank you.
Sunil, you should be able to unmute yourself with the video. You should be able to show your face and introduce yourself to them. Try. Can you try it now? Try to turn on your video. Okay, it's gone, I guess.
And Christian, Rams, let's give Christian a turn. He has been waiting. Christian, are you there?
Yep.
All right. Let's wait for Christian. He might have I don't know. Might have difficulties. I really enjoyed your both talks. It's been really very inspiring, and I have a question for Tessa. I was thinking about the diffusion rate of the protein, especially when you are talking about these growing aggregates, and when they're one of them. To me, kind of makes sense to Yeah, they are all not made like the same, but how do you include the diffusion of the monomer to the aggregates? And my second question is, how do you plan out? Do you plan to factor in the chaperones or other factors which are binding?
Yes, so the question of diffusion is kind of a complicated one. But in principle, for us, it doesn't matter where the monomer is or which monomer it is, or I mean, it's going to be probably oligomers or multiple molecules coming together that are involved in nucleation. But I think diffusion is going to play a role in the growth of the inclusion, because actually we don't yet know what are the mechanisms, but there must be some secondary mechanisms like secondary nucleation or fragmentation, because this inclusion is gigantic, right? So it's composed of many different amyloid fibrils. And so there probably diffusion is going to play a role, because fragments might break, diffuse for a little bit, and then grow again. But it's probably limited because we are seeing quite compact inclusions. But in other systems, this might actually play a role, yes. And then your second question was about chaperones and other factors. So yes, actually C. elegans is a really great model system for that. You can knock down genes with RNAi pretty easily. So we are currently doing that. And also another interesting feature of C. elegans is that you have lifespan mutants. So a single point mutation can really make the lifespan like twice as short or as long. And so we're also very interested to see whether aggregation actually scales with lifespan or it's really an independent process.
Yeah, I meant more like, how do you modeling the binding of the chaperones? And are you just treating it as an inhibitor, or like there's just a little bit logistics behind that?
Yes. So basically the way people do it in vitro is that they just model it as a perturbation to one of the aggregation steps. So for example, you can say that nucleation is decreased or increased by this much, but not explicitly model in somehow the action of the chaperone. So that's what we would first try to do.
Thank you. I see Christian is here. Christian.
Yes, hello. Before I was unable to unmute myself, so the host prevented me from doing that. No, it was just a glitch, a technical glitch, I guess. So I have two, in principle, I have two questions. One is, I remember Brett Hyman was once showing that a beta aggregates in mice, I think with two photon fluorescence microscopy, so that they were coming and going, which I'm not sure whether this is still true, but I understand that in your system you did not see that, but you mentioned also in C. elegans that you had overexpressed a beta, and so of course it could be a protein dependent. So my question is, when you overexpressed a beta in C. elegans, did you see this appearance and disappearance of aggregates?
What do you mean, they get degraded, or what do you mean?
Well, I mean, what Brett Hyman described was that at certain spots, I mean, they were imaging the mice over several days or weeks, and they were observing that aggregates were there at a certain spot, and then maybe the next day they had disappeared, and then maybe they were coming up again and stuff like that. So there was, they appeared and they also disappeared. Okay.
Yeah, I'm not aware of that. So for the polyQ, we don't see that. We did get also this question from the reviewers, whether we see degradation, for example. In principle, not, but you can, of course, imagine that nucleus could actually be degraded if it's fast enough before the aggregate can grow. And so we do not take that into account. It's basically included in our nucleation rate. Okay. Yeah, we are measuring the effective.
I can look up this paper again and just send it to you.
That would be interesting. Thank
you. Other question I had was you mentioned that there was a volume dependence there was some reproducibility in where the aggregation took place, and but then you said, I think, that you had expressed only in the muscle cell walls the the polyQ. So then I was a little bit wondering, I mean, was it not expressed in the other cells as well? Because then, of course, it can only happen there, right? Or, I mean, you mentioned that the nucleation was volume dependent. And
yes. So we are only considering the muscle cells.
Okay.
Right?
And the other cells, they do not change their volume, or?
Yeah, okay, of course. Yeah, the whole worm grows, of course, which is why the muscle cells also grow. I mean, that's part of the process. But we are only expressing the protein in the muscle cells. So that's why we're only considering those.
Okay, and then, I mean, I could imagine that certain muscle cells grow sooner or later, or grow.
No, not really, no, because as I also showed, they are all physiologically identical.
I see.
So all 95 are really exactly the same size also during development. So they grow also at the same rate. And then I also saw a question about this in the Q&A, because it's important to note that also the protein concentration, it's not, so it doesn't get diluted, but actually there's continuously more protein being produced. So we found that even the protein concentration also increases a little bit during development. So that contributes actually even more to this effect. So I didn't have time to show all of that, but yeah, it's actually both. And so what matters in the end is the number of molecules in the system.
Okay, thanks a lot.
Okay, just to say hi to both, because now I can see you guys. It was nice meeting you guys. Very good talk. So I'll see you guys hopefully in the near future somewhere, you know. Thanks. Otherwise, yeah. Bye-bye.
Bye, Sonia.
Bye. Bye.
Great talks, Wenjing and Tessa. There are some interesting questions in Q&A, Magda. I promoted Binzu into the panel.
Oh, okay, yeah, Binzu.
There are other
Sorry,
questions as
I
well.
was not keeping track of this Q&A. We're going to.
There are also some questions.
I think the two of them are from Binzu, and the other ones is if you can check it. Yeah, but Binzu, it's your turn now.
Yes, hi. Very interesting talks. I have a question for each of you. So I think for Tessa first, your model showing the non-cooperative model fits much better with your data than cooperative model. But there are numerous studies in the literature showing the spreading of aggregate protein, to the neighboring cells has been reported. And I'm just wondering whether this is because of your system of study, because you focus on the muscle cell. Maybe those cells doesn't have a lot of, those intercellular endocytosis or exocytosis processes or some other things.
Yeah, so that's a great question. Of course, it could be because of the cell type, it could be because of the protein, or it could be because of the worm. My guess is that it's mostly because of the protein. So again, because this is a simple polyQ protein without flanking regions, and it's probably not being secreted or anything. So there have actually been some studies on alpha-synuclein in the worms, and that actually does spread. When you express a yeast prion protein, also it spreads, and it can actually also escape from the muscle cells. So I don't think it's necessarily because of the muscle cells.
Very interesting. So I think it's more protein dependent.
Yeah, probably. And then, I mean, of course, I also don't know how much communication there is between the muscle cells. So what people found was basically from the muscle cells to other tissues. Mm-hmm. But in the neurons, of course, it's going to be different again, because they're all really like physiologically connected, right? So.
Right, yeah. But it's much more complicated, right? As
yes,
you see in your last part of the
we are working on it. But as you could see from my preliminary data, it looks like a non-cooperative aggregation curve. So, yeah.
Thank you. For Wenjing, so in your screening, yeast display screening, you mutate like a CDR loops of the nanobody. So I think in your slide showing like a 10 or 14, 20 residue changes. So your ultimate, like the best nanobody showing 1 or 2, how many amino acid has been changed compared to the wild type?
So actually the library we created is really large, because there are 3 CDR loops, and we randomize all 3 CDR loops. The first 2, so maybe there are at least like I guess, I think 4 to 7 are totally randomized, and some are like randomized to specific amino acids. And the third group, really with the 10 amino acids, completely randomized. So therefore, there's it's hard to compare to the wild type because it's it's all different, because probably 20 amino acids are like completely randomized. So the sequence space we can cover is really, really large. But the yeast library actually is small, only covers a small space of all the variations. Yeah.
So your final nanobody, the best nanobody is like there are multiple changes in terms of amino acids on the loops.
Right. Yeah, completely different from the original template. Yeah.
Thank you.
No problem.
Magda, there are questions in Q&A. Also, there's a question, 2 questions in chat box.
Yeah, I think the question in the chat box was,
Matthias,
I believe Christian already asked, but the question from, there's anonymous question from, since the protein concentration is constant, increase in cell volume leads to protein dilution. Shouldn't this decrease aggregation? That's the question.
I just answered that question.
Oh, sorry, I was
yeah, don't worry. I tried to answer multiple at the time.
Yeah. So yeah, that's most of the questions, Rams. If you guys see any other which are missing, just let me know.
There are 2 questions in the chat box. One is from Matthias Buck, maybe already answered. Just to make sure that, can you look at them? There's one from Sahao He. Wenjing, can you look
Do you
at
see
them?
the chat box or?
Yeah, chat box. What is the GFP PFF NB2 post positive, but synuclein P129 negative dots in the HEK cells that transfected with synuclein and transduced with synuclein PFF? Do you see that, Wenjing?
Yeah, I saw that. Okay, yeah. So I think Zhuo Shenhe is not here anymore, but I can share the screen to show what he meant. So he meant in this colocalization assay of the nanobody with the phosphorylated alpha-synuclein. So we see, so there's these large aggregates. So the protein is expressed, the GFP is expressed throughout all the cells, and we did notice, I guess, there are a couple, so like these dots here that are, but didn't show up here in the phosphorylation. So it's possible, right? So we don't really know what these are, but they could be possible that maybe the alpha-synuclein is not phosphorylated yet. Right, for this one, we didn't stand against just the alpha-synuclein. We should have, that probably will provide more information on whether maybe actually also has the alpha-synuclein aggregates, but just not phosphorylated yet. Yeah.
Great, great talks. Magda, you can wrap it up.
Yeah, it's it looks we went through all the questions, and thank you everybody for the great discussion and for coming here and sharing your great research and your time, of course. And I don't see any more questions, so just we are ready to go. And